Hello and welcome back. In this 9th video of the series, we are going to analyze the syllabus and the topics of cell biology. Cell biology is the first major area of paper 2, but it is one of the easiest parts of paper 2, and with reasonably less effort, you can hope to get very good marks in cell biology. It is also an interesting part, and last but not the least, it will lay the foundation to better understanding of at least two other areas of paper 2 syllabus, that is genetics and plant physiology. So seen from conceptual point of view, concept building point of view, it is one of the foundational area which will help you get a mastery over other areas, and seen from the examination point of view, it is one of the easy but scoring area. Like all the videos, we are going to take an overview of the syllabus, then we will go for individual topics analysis, and eventually we will see what will be a good source to study cell biology. Let's start our journey of cell biology and let's take a look on the syllabus. So here on the board you have the syllabus. As you can see, there are 25 topics. Topics are fairly specific except perhaps the first topic, that is techniques of cell bio, because the UPSC has not specified what all cell biological techniques that you have to study, and therefore you have to take a look on the past 15 years of question paper so that you can understand what the UPSC has asked from this topic. You don't have to do that job. I have already done that, and I'm going to suggest what are the individual techniques that you should be studying. After the topic 1, if you pause the video and you can take a look on each and every topic of the syllabus, you would notice that every other topic is fairly clearly specified, and that gives you a fairly good idea what kind of question you can expect in the actual main examination. Barring a few topics, the IFS syllabus is identical. In the IFS syllabus, there is just one additional topic, that is Lambrus chromosome. Other than that, in the IFS syllabus and in the civil service syllabus, there is no significant difference. So to these 25 topics, if you add one more topic, Lambrus chromosomes, then you would have prepared cell bio for both civil service main and IFS main examination. On that note, let's start the individual topics analysis, but quickly we will take a look how in paper 2 the relative weightage has been assigned. So I'm going full screen once again so that you can see on this pie chart, and you will notice that how the proportionate weightage has been given. Approximately 16% weightage has been given to cell biology in the last 15 years. This entire exercise of assigning the average is actually the average taken over 15 years of UPSC main examination. Cell bio gets 16% of question weightage, which is a very good weightage to have. The second largest area is physiology, that is marked in orange color. That gets exactly double the weightage, but we have to realize that physiology is a big area. But the largest area of paper 2 is a combined sphere of 4 topics, that is genetics, plant breeding, biotechnology, and biostatistics. These are in a way related parts, and therefore I am treating them as one big unit, and this big unit, as you can see in the graph, is getting 40% weightage. The remaining 12% weightage has gone to ecology and plant geography. So that is the weightage that has been given to different parts of paper 2, and here we are right now going to look into cell biology, which gets 16% weightage. When we talk about 16% weightage, it means cell bio minimum gives you 40 marks of questions, and there have been years when it gives you up to 60 marks of questions out of 250. So the year when it has been given 60 marks, the weightage goes up to 25%, but that has not been year after year. Therefore, the average comes down to 16%, but 16% also is a sizable weightage to assign to any given topic, and you should be taking this part rather seriously. Now let us take a look on individual topics of cell bio and see what kind of questions may be asked to you, what are the subtopics that you should be doing in cell biology. Here we go with the very first topic, techniques of cell biology. Here you have to do 3 specific techniques and 1 overview of the techniques to study eukaryotic cells. So fourth topic that is mentioned here is essentially an overview that you can prepare in approximately 300 words, and it will include everything: isolation to fractionation, separation of different cell components, so on, and then of course an overview of microscopy. But this kind of overview question has not been asked in the last 15 years. Only once it was asked about 18 years back. It was a short note, and most of the candidates who had even a basic idea of the basic cell biological technique did not struggle to write this answer. If you see the past few years question, only these 3 techniques, electron microscopy, structure and working principle of compound microscope, and phase contrast microscope, they have been asked. You should prepare each one of them in approximately 250 words, each one in 250 words, plus you should draw clear line diagrams. Whenever you are describing a microscope, line diagrams play a very important role. That line diagram has to come. Once you are done with the technique, the next topic is going to be the prokaryotic and eukaryotic cells. Here you do not have to do in detail the structure of prokaryotic cells, because this is something you have already done in the beginning of microbiology. There is no point repeating it. If at all structure of a prokaryotic cell is asked to you, you will be very well be in a position to answer this question because you have studied it in microbiology. No effort is to be taken for that. What you should do here is to establish a clear tabular difference between prokaryotic and eukaryotic cells. This difference has been asked and can be asked. One tip I would like to give you to score well in this question: within the table itself, if you can draw small diagrams to highlight the structural parts that you are talking about or to highlight the structural differences, it will really be nice for the examiner to see your conceptual understanding. And such tabular differentiation containing a small diagram, they add a good value in comparison to your competitors, because most of your competitors are perhaps not going to take this effort to insert a small structural diagrams. So it is a step well worth taking, and it will have an impact upon your score. Next topic is the extracellular matrix and the plant cell wall. When we look at the plant cell wall, it's a reasonably small topic. There are fairly clearly defined parts. First, composition of cell wall. So here you have to do the microfibril components, the matrical components, and the cementing material. Here you also need to be updated in your content, because most of the conventional Indian books do not give you an idea that plant cell wall also contains certain structural proteins, certain elasticity regulating proteins. But that part is a very important aspect of plant cell wall structure, plant cell wall composition. When you read from good books, and in the end I have suggested those books, you will be having all this input. So it's not a very rare piece of information. You can easily get this information. I'm suggesting it so that while covering this topic, it comes to your mind that, okay, I have to do this also, and that will be taken care of. Next one is ultrastructure of plant cell wall. Plant cell wall, as we see from the electron microscope, that's what you have to do. Molecular structure of the cell wall will include how the different components of cell wall, that is cellulose, microfibrils, hemicelluloses, pectin materials, and structural proteins. These are the 4 components. How do they associate with each other? And they give rise to the 3-dimensional structure of a plant cell wall. That's molecular structure of cell wall. The 4th thing that you have to do is to describe properly primary, secondary, and tertiary walls of plant. This 4th question is basically a reworded or differently structured question number 2 only. So when you are dealing with the ultrastructure of the plant cell wall, you anyway come to describe primary, secondary, and tertiary wall. So this question is another way of asking question number 2. So when you are preparing a short note, prepare it as one short note and be ready to answer this, whether it is asked as an ultrastructure or it is asked as the primary, secondary, and tertiary wall. Lastly, you should be doing the functional role of plant cell wall, how it is protective, how it provides plant tissue a proper structure, how it keeps a plant cell in the context of the neighboring tissues, how does it allow certain material, so on. What role it plays in shape determination of the plant cell, what role does it play in maintaining the water relation of the plant cell. So these are some of the structural aspects that you will be dealing with. The next topic is going to be cell adhesion, where you will be dealing with plasmodesmata as well as the middle lamella, which is the cementing material between 2 adjacent plant cells. So if you see, first and foremost, ultrastructure and functions of plasmodesmata you should be doing. Best coverage of plasmodesmata is given in plant physiology textbook. By Taze and Zeiger. You do not find this good a coverage in any of cell bio book. So especially for plasmodesmata, you should be studying the first chapter of Taze and Zeiger. There it will give you a very good coverage of plasmodesmata. What is the ultrastructure, how it is formed, what are the types, so on. Next, you will be dealing with the middle lamella. Here, plant cell adhesion and molecules and structures responsible for that. And molecules basically include different pectins, and the structure is called middle lamella, that you should be doing well. Now the time to do membranes. In membranes, basically there are 2 parts. First part is composition of plasma membrane. We all know that plasma membrane is made up of lipid bilayer, sterols, sphingolipids. Then it has got integral, peripheral, and anchored proteins. And finally, it has got conjugated oligosaccharide. Oligosaccharides are always conjugated. That means it is either attached to membrane lipid or it is attached to membrane protein. That's the composition. In the composition answer, you will be giving a small detail of each constituent of plasma membrane and what is their role. So proteins are basically responsible for providing the functional features to the membrane. On the other hand, the lipid bilayer is responsible for giving a structure to the membrane. Carbohydrates are also related to the functional aspects of membrane, such as cell-to-cell recognition, signal reception, cell-to-cell adhesion, so on. Each one needs to be biochemically described and each one's role also needs to be described. And do not forget to mention their subtypes, such as lipids. So they have 3 types: phospholipids, sterols, and sphingolipids. Proteins, they are based on how they associate with the lipid bilayer. So you have integral, peripheral, and anchored protein. Then there is further subtypification. If you are reading from book like CARP or any foreign author's book, in the current edition, you will get subtypification of integral protein, subtypification of anchored protein. Pay attention to that. The second part of membrane is going to be the ultrastructure of the membrane. So fluid mosaic model has to be discussed. And apart from the fluid mosaic model, in the last 20 years, there have been a number of modifications in the fluid mosaic model. To be clear, no part of the fluid mosaic model has been contradicted by the scientist. ऐसा नहीं है that any part of the fluid mosaic model is no longer valid. Fluid mosaic model given by Singer and Nicholson is still the most valid explanation of the membrane structure, but there have been a lot of new insight which have been added to the fluid mosaic model. That's what you have to cover. A good book covers that, and in the end, I'm anyway going to suggest. The next topic is membrane transport. Membrane transport is a conceptually a small topic, but many questions are possible from membrane transport. Therefore, it looks like a big topic. These are the things that you should be doing. First, do in detail the modes of passive transport, then the modes of active transport. After that, difference between passive and active transport mechanism, then difference between primary active and secondary active transport, then difference between ion channels and pumps, and difference between ion channels and transporters. Each one you should prepare, each one, in about 250 words. And in my personal experience, I have noticed that membrane transport topic is not well covered in the Indian textbooks. So I would suggest the international textbook, preferably Gerald Karp's book, is an excellent source to do majority of the topics of cell biology. So you can stick to that book and it will give you ample conceptual insight and very high quality facts, updated ones, to help build your concept and information base. Moving to the next topic, once we are done with these, you will be coming to vesicular transport. So vesicular transport is when macromolecules move from inside the cell from one organelle to another, or from inside the cell to cell secretion, or from outside the cell to the interior of the cell through endocytosis. All of these transports, and of course phagocytosis also, they take place by vesicular transport mechanism. In vesicular transport mechanism, endoplasmic reticulum, Golgi apparatus, and plasma membrane, they all can act as the donor of vesicle membrane, and at the same time they can act as destination for vesicular transport. I would suggest that this topic should be done ideally after doing endoplasmic reticulum and Golgi apparatus. Once these two topics are done by you, then you should be doing vesicular transport. But when you do vesicular transport, the first and most commonly asked question in vesicular transport is an overview kind of question that gives an account of vesicular transport, its mechanism and its significance. Additionally, you should be doing secretory pathway and go in some detail where you will be learning about coat protein, two coated vesicles, clathrin coated vesicles, so on. And then also do endocytic pathway. Endocytic pathway is anyway an important topic to be done in the context of endosomes, which is the topic just before lysosome. Next topic is going to be chloroplast. The subheadings for chloroplast and the subheadings for mitochondria are nearly identical. Pay attention to these. First, you should do ultrastructure and functions of chloroplast. This topic is better given in plant physio book. So I would say here that rather than depending on a cell biology book, you should depend upon a plant physiology book. After doing the general ultrastructure of chloroplast, get into the ultrastructure and function of thylakoid membrane and the four complexes which are present in the thylakoid membrane. Do it in some detail. It is PS2, PS1, cytochrome B6F complex and ATP synthase complex. This is a very important question that has been often asked in the main examination. And finally, you should do endosymbiotic origin of chloroplast and how primary chloroplast evolution took place and how secondary chloroplast evolution took place. Origin of chloroplast has routinely been asked in the main examination, although less frequently in the last 10 years, but if you broaden your horizon, you will notice that it is one of the questions that UPSC has asked. Exactly same subheadings you have to apply for mitochondria if you see ultrastructure and functions, then the inner membrane of mitochondria ultrastructure plus the 5 complexes of inner membrane. What are the 5 complexes of inner membrane? They are known as complex 1, complex 2, complex 3, complex 4, and mitochondrial ATP synthase complex. Complex 4 is NADH Q oxidoreductase. Complex 2 is succinate dehydrogenase. Complex 3 is Q cytochrome C oxidoreductase, and this one is cytochrome C oxidase. Each complex needs to be explained by you, and of course you have to do in detail the ultrastructure of the inner membrane, because functionally this part of mitochondria is extremely important. And similarly, because mitochondria are organelles of endosymbiotic origin, so you need to know about the origin and evidence for this origin for mitochondria as well. Once you have handled all of these in approximately 200 words plus diagrams, you are done with this topic as well. Now you will be coming to endoplasmic reticulum. In endoplasmic reticulum, a general account of ultrastructure and functions of ER, then difference between smooth ER and rough endoplasmic reticulum. Make sure that you are differentiating between them not only on the structural basis but also on the basis of their biochemistry, their internal luminal activities. Then signal hypothesis related to protein import. This needs to be done. There are steps which you will be describing with help of diagram. And lastly, the secretable proteins or lysosomal proteins, their processing starts within the ER lumen. So protein elaboration for such proteins which are destined to go outside the cell or in the lysosome, that elaboration starts in the lumen of endoplasmic reticulum that we need to do. So protein elaboration in ER. These are the 4 topics you need to do. And then you come to dictyosomes or Golgi apparatus. Again, general account of Golgi apparatus ultrastructure and functions. Prepare the difference between Golgi apparatus and endoplasmic reticulum. And here again, both structure and functional difference needs to be established. And lastly, you will be doing protein elaboration in the Golgi apparatus. The protein elaboration that starts in the ER is completed within the Golgi apparatus. So how protein is elaborated in Golgi apparatus is also something that you need to know. Even if it is not asked directly, it will be an important point to establish distinction between Golgi and endoplasmic reticulum. So the next topic is ribosomes. It's a small topic, and after this also there are a few more small topics which will be coming your way. So in ribosomes, first general account that you should prepare is structure, ultrastructure and functions of ribosome. In ultrastructure, you will do the subunits, then 3D structure, and then the domains of ribosome. And by function we mean the role of ribosome in translation. This should be prepared in approximately 300 words, and this is the most likely question to be asked to you. And then approximately 150 words, you should do the James Lake model of ribosome subunit assembly. This model tells us that how at three-dimensional level the ribosomal subunits interact with each other and form a definite 3D structure. It can be asked as a tail, and long back it had been asked as a standalone short note in the Forest Service main examination. Next question is endosome. Just one thing that you need to prepare, that is ultrastructure and function of endosomes, and add a tail on their position in the endocytic pathway. You should be preparing it for 250 words, and this part will also give you an idea what is their relation with lysosomes. And next topic is logically lysosomes. Here ultrastructure and functions of lysosomes needs to be done. A critical analysis of lysosomes as suicide bags needs to be done. Does it really deserve to be called suicide bag or not? That is for you to find out, but I'll give you an idea that lysosomes play no primary role in programmed cellular death. Therefore, the modern day cell biologists, they hesitate in calling the lysosomes as the suicide bag of the cell. Reasons you will find when you study it. And then you have got enzymes of lysosomes. So what are the major categories of the enzymes that we find in the lysosome, and how these enzymes are responsible for lysosomal function that you need to do. And finally, you need to do the types of lysosomes. The types of lysosome will basically involve the primary, secondary and tertiary kinds of lysosome. How are they interrelated, that you need to do. The functions of lysosome you have already done. So with this, you will be completing lysosome. Next topic happens to be peroxisome. There are two things. One, you should be preparing a general account of what's peroxisome. Its structure is very simple. So what is the structure? And finally, what is the biochemical role of peroxisome in cell metabolism? And second, in the plants, a modified type of peroxisome is found, and that is known as glyoxysome. Glyoxysome is present in many different cell types, not all the plant cells, but in many different cell types. And one of the cell types is concerned with oil storage in oil seeds. Think of mustard seed. So in mustard seed, the reserve food is present as oil. But when the seed germinates, the germinating seed requires sugar. So what is the pathway that converts the stored oil into sugar? It is in a way a gluconeogenesis type of pathway, but it starts with something known as glyoxylate pathway. And the modified peroxisomes where the glyoxylate pathway takes place is known as glyoxysomes. So that you have to do. So these are the two things. If you do on peroxisome, you will be pretty sufficiently covering this topic. Then you come to nucleus, chromosome, etc. So the rest of the topics, they hover around the nucleus, chromosome, genome packaging, and then you will be coming to cell division and signaling, etc. Let's take a look. Next topic is, okay, I forgot a small topic, that's cytoskeleton and microtubule. In cytoskeleton and microtubule, first a general account of cytoskeleton and cytoskeletal elements of plant cells, approximately 250 words. Then specifically you should prepare on microtubules, what is their structure, how they are important in mitosis, how they are important in determining the cell shape, in vesicular transport, keeping the organelles in their right place, so on. After that, you will also be doing the ultrastructure of eukaryotic flagella. Sometimes it will help you in paper 1 when they will ask you a description of algal flagella. So whether the algal flagellum is tinsel type or whiplash type, internally it has the same 9+2 microtubular organization. So you should be knowing ultrastructure of eukaryotic flagellum here, after doing microtubules, and then you will be easily answering the ultrastructure of algal flagella in paper 1 also. So these are the 3 topics that you should be doing in cytoskeleton. There is no need to study intermediate filaments and microfilaments in great detail. Microtubules need to be studied really well. After that, nucleus, nucleolus, and nuclear pore complex. 3 things you have to do independently: structure and functions of the nucleus, ultrastructure and functions of the nucleolus. Here you will be dealing with the ribosome biogenesis, the different locations where ribosomal RNA are transcribed, so on. And finally, ultrastructure and functions of the nuclear pore complex with proper diagram. After doing that, you should do chromatin and nucleosomes. Again, very straight coverage you are expected to do. Molecular structure of chromatin fiber, and when you do this, so you have to describe the solenoid and zigzag model both, and then you should be coming to even finer level of a structure, that is the composition and molecular structure of nucleosomes. Here you will be dealing with how histones interact with DNA, how the linker histone, that is histone 1, plays a role in stabilizing the entire structure, define the terms, give the dimensions correctly. These are the basic requirements to answer this. Any good cell bio book will be doing a very good coverage of these topics. Moving next, you have a topic, chromatin organization and packaging of genome. Here you should be preparing euchromatin and heterochromatin very well. And then heterochromatization, that is the mechanisms by which heterochromatin is formed, and types of heterochromatin, that will essentially imply constitutive heterochromatin and facultative heterochromatin. So this will be the genome packaging and chromatin organization, and then you will be coming to cell signaling. Cell signaling in cell bio book is a very big topic, but in the UPSC Botany paper 2, the requirements are very specific, so you should remain confined to that. There is no need to overburden yourself. First, an overview of cell signaling. This is a very common question. Prepare it in approximately 300 words. After that, cell receptors. What are the major types of cell receptors, and what is the mechanism of action? In a tabular format, approximately 300 words. Here you will also need some diagrams, and here you will need mainly flowcharts. After that, you need to come to details of G protein mediated signaling. So you need to know the structure of G protein couple receptor, then the structure of heterotrimeric G protein, and of course the mechanism of action, how the receptor is activated, how the G alpha subunit is activated, and then what happens when the G alpha subunit is activated. Lastly, the role of cyclic AMP as a second messenger. These are the 4 topics you should be doing in cell signaling, cell receptors, and it will be taken care of. Next one is mitosis and meiosis. First, do very well the stages of mitosis and make it more plant cell specific. So here you can again take a look into plant physiology textbook, because in plant physiology textbooks, the cell division is given more with the with reference to the plant cells, so that will be a good coverage. And then you should do meiosis in detail. Meiosis is very nicely given generally in genetics books, but even in cell bio book like CARP, it is wonderfully given. So do stages of meiosis. And then come to the details of meiosis prophase 1. That's a separate question in itself, because this is a very prolonged part of meiosis, and this is where the crossing over also takes place. So it's very, very significant both from the cell division point of view and for you from the examination point of view. And last question is difference between mitosis and meiosis. That's also an important and often asked question, but it is a very simple one. Once you have done mitosis and meiosis well, this is not a difference that will cause any problem to write. Next one is molecular basis of cell cycle. In molecular basis of cell cycle, first molecular mechanism of mitosis should be done. That means how spindle fibers are formed, how do they elongate, how do they carry out chromosome capture, how do they stabilize during the metaphase, what causes the separation of the sister chromatids, what causes the shortening of spindle fibers during the anaphase, so on. After that, the regulation of cell cycle needs to be done. What is cell cycle regulation? Why cell cycle regulations should be done? What are the molecular constituents of cell cycle regulation? And what are the steps of eukaryotic cell cycle regulation? My suggestion will be that do this topic with a specific reference to Saccharomyces cerevisiae, because in plants, this model of cell cycle regulation fairly closely applies. Lastly, you have got cyclins and CDK and their role in cell cycle. So these are the topics that you need to do. After that, you will be coming to a topic that is more of a genetics topic, and no good cell bio book has a coverage of this, but in proper genetics book, there is a very nice coverage. So this is something, in my opinion, you should be looking into a genetics book. Later, when I suggest a book for genetics, my suggestion will be Klug and Cummings. So from Klug and Cummings, you should be doing different types of structural variations in chromosomes and their outcome, then different types of numerical variations in chromosome and their outcome, and from a structural variation, oftentimes they have asked a short note on chromosomal translocation, so that you should prepare as a separate question. But as I said, there will be no good coverage in cell bio book. For this, you will have to study a genetics textbook. Next is polytene chromosome. So it's fairly straight: ultrastructure, properties and importance of polytene chromosome. In the IFS, you also have to do lamp brush chromosome. So there also you have to do ultrastructure, properties and importance. And same will go for the last topic of cell bio, that is B chromosome. These are accessory or supernumerary chromosomes. There also you have to do ultrastructure, properties and significance. In the end, what should you study for cell bio? My strong recommendation is that if you are wanting to study a textbook, because you have an option to study from my study material, my book on cell bio, but if you want to study from a textbook, I will very highly recommend the book on cell and molecular biology by Gerald Karp. It is very simply written book, but at the same time, information wise, it is very, very rich. Even in my own book on cell bio that you see on the right extremity, I have taken a lot of content from Karp and other books, of course, like Lodish, Alberts, Cooper, so on. And if you are someone who prefers an Indian book, then amongst the Indian books, I would suggest that P.S. Verma's book is actually one of the better written Indian books. Plus, this book gives you a very decent coverage of genetics, ecology and evolutionary biology. So one book covers many areas. So you can take this one book. You will find it helpful in other parts of the syllabus also. So this is my recommendation for what you should study on cell bio. So now this is your time to pick up one of these resources and start covering cell bio as I have advised to you. This should not take you much effort or much time, but it will be an effort well spent. It will give you very nice rewards in paper 2. So on that note, I will sign off for the day. Good luck and goodbye to all of you. And I hope you keep working hard and progress purposefully towards your journey to success. Goodbye.